Hello and welcome. My name is Dr. Brian Hill. I'm the director of the Lymphoid Malignancies Program at the Cleveland Clinic Taussig Cancer Institute. I'm joined today by Dr. Jennifer Woyach, who is the section head of the CLL program at the James Cancer Center at Ohio State University Medical Center and the co-director of the Leukemia Research Program. We're going to be talking about two different abstracts that were presented at the recent 2022 American Society of Hematology meeting. And welcome, Dr. Woyach.
Hi, Brian. Happy to be here.
So we'll start by you were involved in a couple of different, many different projects, but the couple that we were going to discuss today was the first one is more of a sort of a preclinical effort targeting Venetoclax-Resistant using CLL with a PROTAC-based BCL2 BCL-XL degrader. So maybe you could just tell us about that.
Yeah, absolutely. So this is actually a project that is primarily led by Dr. Deepa Sampath at the MD Anderson Cancer Center with a graduate student of hers, Daisy Rohina. And in this project, we were looking at a BCL2 BCL-XL degrader that was initially developed by two scientists at the University of Florida. And so the reason for looking at this is we know that many patients who are on Venetoclax, especially those who have relapsed refractory CLL, will eventually develop resistance to the drug. So definitely a clinical need for our patients. And one of the paradigms that has been really exciting over the past few years in preclinical and now clinical development are these PROTACs. And so a PROTAC is a proteolysis-inducing chimera. And basically it is a molecule that has two parts to it. One part will bind to the target, and then the other part binds to something called an E3 ligase, which targets a protein for degradation inside a cell. So basically the concept is you target something and then you degrade it. And this is used clinically in CLL in trials with BTK degraders. And so here we were interested to look at a degrader of two proteins, BCL2, which is the target of Venetoclax, and BCL-XL, which is the target of another drug called Venetoclax. And BCL-XL degraders have been limited therapeutically because they cause platelet toxicity. But the idea is maybe if you were to be a little bit more targeted in your approach, targeting specifically leukemia cells, and if it was more of a not super potent degrader of BCL-XL, you may be able to get around some of the platelet toxicity. So this drug we tested primarily in vitro using primary CLL cells, using models of Venetoclax resistance, where we put in mutations in BCL2 that we know induce resistance to Venetoclax. And we were able to show that this PROTAC degrader can cause cell death in all of these models. And it really looks very exciting, including in many models of Venetoclax resistance. This drug is actually being studied in acute myeloid leukemia as well. And so the hope is actually we may be able to take this drug to the clinic in the next few years in CLL.
So this is really an exciting, I think, really novel approach to targeting sort of intracellular targets. And the BTK degraders that have been studied, I think, are the first in class of this sort of novel potential whole new area of therapeutics. Where is this one in terms of maybe moving towards phase 1 human trials?
I think that this one is purely preclinical for right now, but the makers of the inventors of the drug are really interested in moving it forward. Our next step is going to be to look at it in some mouse models of CLL, including mouse models of Venetoclax resistance, and hopefully that'll provide some justification that you might see efficacy in patients with CLL.
Yeah, no, what's remarkable is how fast the pace of discovery has been in recent years, going from preclinical models and now, as you point out, we have known mechanisms, at least of some of the things that may contribute to Venetoclax resistance, including these point mutations in BCL2. So that's exciting to hear about, and glad that that work is ongoing.